people, we are live and people are starting to pile into the room. There are probably, I don't know, I'll get a number count soon, but we got, I see a bunch of people in the comments section. Welcome everyone. Nick Money, thank you for being a sport and joining us this morning. So your Fungal Consciousness paper, what was it, a paper or what would you call that?
It was a message to humanity. Your
message to humanity has been received, and people have been talking about it, and today you're going to give a presentation on fungal consciousness. So the floor is yours.
Okay, well hopefully I don't see the number of international guests out there, but I'm assuming indeed it is a...
And people, just let us know where you're tuning in from. I'll start to push it up onto the screen.
A vast sea of individuals, but
yes.
All right, well thank you, Peter, and I guess we've struggled through the technical issues here. Always makes me think of Ernest Hemingway and his famous quote about grace under pressure. He was talking about his service as a journalist in the Second War. But then, of course, he was shit-faced at the time, no doubt.
So you see this? We got people from Las Vegas, SoCal, Pennsylvania, Chicago, San Diego, Boston.
Okay. Good.
We got some Brazilians in the house. Denver. Right. Whoops, these are going by fast. New York. We got Great Britain. We got Eureka, California, Washington State, NorCal as well, another UK. We got Maine. And where are you originally from?
I grew up in, and thanks for interrupting my anecdote about Ernest Hemingway, but there you go, it wasn't. Anyway, he was dealing with technical issues during the Second War. We're dealing with, issues with streaming webinar issues. So anyway, and of course he was shit-faced at the time, which I'm not.
The day is young, though. You could change that.
It's true. Although I did quit drinking recently. I'm one of those pandemic cases of pandemic austerity, puritanism. So.
Anyway. Of all the times to quit drinking, I don't think during the pandemic.
I did so much of it early in the pandemic. I think it was time. Time to move on. Where were we? I was born in Oxford, England, in 1962. So I'll be 60 in May, which is beyond hideous, as is revealed with this camera. So I'm off for cosmetic surgery as soon as we're done here. But yeah, I've lived in the United States since 1986. And I've worked in many places on the East Coast, you name it, Colorado for many years, and I've been in Ohio, which is a surprisingly wonderful place to live, other than the political atmosphere, for the last 25 years.
And what got you into mycology in the first place? I mean, were you kind of, when you were young, were you always into botany and plants and nature and...
No, I was fortunate that my grandmother on my mom's side was a wonderful natural historian. She reveled in nature. She had no formal education in biology, but she was just the most wonderful, glorious woman. And she took my sister and myself outdoors, whether or not we wanted to accompany her. And she was my introduction to the natural world. But I had a great teacher. I went to Bristol University in southwest of England, and had a wonderful professor there called Mike Madeline, who was a mycologist, and I fell in love with him and the subject. And now, decades later, I'm still interested in the fungi a bit.
You're still doing it.
I'm interested in lots of things, but the fungi still capture some of my attention.
Yeah, if you're willing after this round to do another one, I'd love to do a deep dive into yeast, and that looks like a fun one too.
Yeah, absolutely. And our own terrifically awful species, Homo sapiens. But actually, the fungi do tell us a lot about our own insignificance in the universe. And so that is partly why they still interest me. I think they have a lot to teach us, or rather, we can learn a great deal from them.
Got it. Well, you ready? You ready to begin?
Yeah. So I guess I've got to say next slide, right? Since you're controlling the
Right.
slide. Yeah.
So let me cue it up, and all right, give me 1 second. This is going to be trippy for a second. Let me just go to Keynote and play. All right, so I'm not going to be able to see anything, so everyone watching, all I see is the slide on my screen, but take it away.
All right. And what's that little bar at the bottom? Does that go away if you?
No, hold on.
We have to go for aesthetic purity. There you go. Look at that. All right. So yeah, I probably got what 30 or 40 minutes worth of slides here. I don't want to bore anybody, but of course you.
So just to give you some context, this is an audience that's appreciative of the deep dive. So don't feel like you need to be fast and gloss over stuff. People, I love the idea of long form content and allowing people to kind of articulate an entire point versus feeling rushed. So,
okay. All right, very good. All right, so fungal consciousness. Here's a nice picture to begin with. Robert Hooke, 1665. It's a drawing of mucor, or a relative, so it's a microscopic fungus, and he found this growing on a sheepskin book cover, vellum, I guess. And this is actually one of the earliest images, published images of any microorganism. So Hooke was first, and he chose to illustrate this fungus, and then another one that's a rust fungus called Phragmidium that grows on this particular species that he pictured was growing on the leaves of a rose bush. So it's interesting in itself. In his time of his plague, 1665, Hooke's Micrographia, glorious book. First images of fungi, first microscopic fungi, first images of any microorganism. And I'm going to talk about the consciousness of these organisms. And I'm not going to use the word organism again, of fungi in this presentation. So if you'd like to go to the next slide. Fantastic. Look at that. It's pulsing. So what we're looking at here, this little GIF comes from a friend of mine, Patrick Hickey, who lives in Edinburgh in Scotland. And Patrick's done over the years amazing things in terms of imaging fungi. And he worked with a good friend of mine, the late mycologist Nick Reed, who died last year. So they produce some of the most glorious images of microscopic fungi ever. And this image here, this pulsing, is actually a time-lapse image, and it shows colonies of honey mushroom, species of Armillaria, that happen to be bioluminescent. And so this is speeded up here. It isn't pulsing with that, what have we got here, heart rate here. This is pulsing up maybe 60 beats a minute, something like that, a bit less. But indeed, there is a periodicity to this in the way that this fungus bioluminesces, and there are changes in the intensity of the light emission over the course of minutes. Let's go on to the next slide. So, shaggy ink cap here. I've been interested in fungi for a long time. And some of my research has involved looking at mechanisms of spore discharge. So when you look at a mushroom, what you're looking at is a sex organ. It's a reproductive organ that produces and releases tremendous numbers of spores. If we flip over to the next slide, we'll see the underside of a mushroom. And you can see the gills. And so the gills are the tissue surfaces over which the spores are produced and released. And mushrooms release immense numbers of spores. If we look at the next picture, See here, if you get the illumination right beneath any mushroom, you can use the lamp on your cell phone. You can pick up this fairy dust that's emitted from a mushroom. About 30,000, in some species anyway, 30,000 microscopic particles per second. So that represents billions over the course of a couple of days. Vast numbers of spores are released from mushrooms. This is their purpose. This is what they do in terms of sending the fungal genes down the great stream of time. So we think a lot about mushrooms whenever most people do, when fungi are mentioned. In a sense, they're the least interesting part of the organism from the perspective of perception, consciousness on the part of the fungus itself, because it's a rather unresponsive, it is a very unresponsive platform that just does the job of spore release. It's a smoke machine. Once it's produced, it operates according to biomechanical principles. Mushrooms are not very responsive to their environment once they form. So instead, what I want to talk about for the next few minutes then is the feeding stage of the fungus, the mushroom colony or mycelium, which consists, and I think this is shown in the next slide, microscopic threads. This is a beautiful image, a scanning electron micrograph produced by the aforementioned colleague of mine, Nick Reed. So he worked in Edinburgh for many years. I met him when I was a student in Bristol. And then Nick Reed went to work in Manchester before his untimely demise. But as I said, he produced these glorious pictures of fungi. So what we're looking at here are spores that have germinated, and we're just looking really at a few minutes of growth. And so you can see the lumps there, the more spheroidal structures are the spores, and they're putting out germ tubes or young hyphae, these young filaments that eventually will develop to form a larger colony or mycelium. And if you look carefully at the middle of this picture, actually you can look around at the top left too, you can see that the colony is interconnected, that individual cells within this structure are forming bridges. If we look at the next picture, this is a classic image from the 1930s by the great Arthur Henry Reginald Buller. He was the Einstein of mycology, if there ever was one. And he was the individual responsible for figuring out that 30,000 spores a second were released from mushrooms. And he also produced this clear image of a young colony of indeed an inkcap mushroom. You see the spore in the middle, and then the filaments that are spreading out from that central point in all directions. And we're just looking at a two-dimensional view of the colony here, because in nature, the colony also penetrates the surface. And so we end up with a three-dimensional structure in rotting wood or in soil. All of the action in terms of sensitivity and irritability, from which we can see the beginnings, the origins of consciousness, or the simplest forms of consciousness in nature, exist here in the colony. Let's have a look at the next picture. I think Peter's disappeared. So I have to describe the next picture for you. There we go. So this isn't a fungus at all. It's a slime mold. It's a thing called Physarum or Physarum, some people say. So it's actually a member of the amoebozoa. It's more closely related to the classic single-celled organism, Amoeba proteus, than it is to fungi. It's very, very distantly related to the fungi. So slime molds are not fungi. Superficially, this slime mold has a structure that we called, superficially, it looks like a mushroom mycelium in a sense, that we've got a central point here and the organism spreading from that point source. We refer to this as a plasmodium because there's so much interconnectedness here. And the structure actually does pulse in real time. Every few seconds, we see this movement of fluid through the tubes of the colony or plasmodium of this structure. What's happening here is that the organism is in search of food. So in this sort of, I suppose it is an experiment here, it's just getting the thing to grow. We've got a central point from which the slime mold has expanded, and it's moved on from the food source that it was given initially to these oat flakes on the outside here, toward the outside of this culture dish. And you can see that the thing has moved in this sort of deltaic fashion, spreading out. And then as it finds that food, then it concentrates its activities, what we call shuttle streaming. It's streaming cytoplasm towards those food sources. The organism actually, in a sense, doesn't eat the oat flakes directly. It's feeding on bacteria within the oat flakes. So amoebae, including this slime mold, feed on bacteria. Now, the reason I'm showing you this is that most of the experiments that have been performed on sort of non-human, simple forms of consciousness have been conducted over the course of quite a few decades now, mostly by Japanese researchers in Japan, on this slime mold. And well, we've gone to a different screen. We're losing everything. Everything's going. Peter is in the stream with audio only, but Peter, you've lost us on the screen. At least I don't see the slime mold anymore. Oh, we're back on that. If you could go to the next slide. Yep. So this shows one of these lovely experiments on slime molds that we might suggest as evidence of consciousness. So what's happened here is we've got the slime mold growing on food sources on the bottom part of this picture, and they're moving across those things that look like ice cubes, but those are just blocks of agar or gelatin, and they've been infused with sodium chloride, lots of salt. And slime molds don't like lots of salt. It dehydrates them. And so this is an obstacle, but they've got to cross that obstacle if they're going to reach the new food, the unexploited food that's at the top there. So you got those 3 discs that contain it's an oat medium, oat flake medium, full of bacteria too. So the slime mold wants, being anthropomorphic here, to cross that salt bridge. So eventually when it runs out of food, it says, screw it, I'm going to go for it, without that internal conversation in all of its sophistication there. But it does indeed cross the salt bridge and moves to the new food material. One of the things that's interesting about this is that if you test the same slime mold in a subsequent experiment, it doesn't take as long to make this decision. It senses the salt, moves across the salt bridge to get to the food. It's actually acting in a predictive fashion. There's evidence of memory there, a simple form of memory. But more than that, the slime mold can actually transfer that information to adjacent colonies. So in subsequent to this picture here, but don't go to the next slide, it'll actually it transmits that information to those adjacent colonies or plasmodia, and then they'll cross the salt bridge also. So this is a simple form of short-term memory. It's also showing habituation to this repellent, and it's capable of transmitting information to its peers. So as I said, scientists have studied slime molds for a number of years. If there's anything new in what I wrote in the essay that was published in Fungal Biology, it was an attempt to widen this discourse and begin to think about fungi, because fungi have not been studied from the perspective of consciousness previously. The experiments have been done for a long time. The addition that I've made through this essay then is to suggest that fungi exhibit simple forms of consciousness, and we need to begin to start this thinking about fungal behavior in terms of consciousness and we apply the term ethology to the study of animal behavior, we need to think about this also, widening this to other organisms. And since I'm a mycologist, I wanted to think about this in terms of fungal ethology. So let's look at the evidence such as it is for fungal sensitivity, irritability, and consciousness. What's the evidence for the fungal mind? Let's look at the next slide. Pretty picture here of a single hypha. And for many years in my research career, I studied the cell biology of individual hyphae. So these are the individual, or these are the filaments that form the larger colony. And you can see this cell has extended, towards us, the viewers here, and it's popped out a couple of branches during its growth. If we look at what's going on inside that cell, we can go to the next picture in a moment. If we go to the next picture, the next slide, this is an image that's based upon, so it's fluorescence light microscopy here. And what we're looking at is, I've mentioned Patrick and Nick before because I'm using lots of their images in this presentation because they are so glorious. We've got this particular hypha here that's been stained with dyes that are connected to particular proteins inside the fungal cell. So we've got the red coloration represents membrane activity, and we've also got this green stain that's indicating it's a particular protein called Rab5. What's most interesting about the green staining is the area in between. You've got these black holes in between. Well, those represent negative stains for nuclei. The nuclei that carry the genetic information in the fungal cell, those are not lighting up. So wherever you see these dark areas in between, we're seeing here the nuclei through negative staining. Let's look at the next slide, which is, there we go. So we got a little series of images here from a video recording. Yep, it is repeating. We're just seeing a lot of activity in there. Lots of things moving around. And indeed, this thing is advancing at roughly this speed. This is almost natural time here. Immense amount of activity going on within that cell. Continuous flow of materials and continuous flow of information is what I'm going to talk about shortly here. Let's go to the next picture, which is going back in time to the 1960s. Some fantastic early microscopy and biochemistry was carried out in the 1960s. The top picture is a drawing. It's based upon electron micrographs that show the structures inside the cell that we were just looking at. So we've got this collection of vesicles. These are membrane-bound, tiny membrane-bound spheres in the tip of the cell. And then behind this, you can see these mitochondria and networks of membranes. You've got the Golgi apparatus, or the fungal equivalent of that structure, and other organelles. There's just a lot of stuff in there. What happens as the cell grows is that membranes are brought toward the tip where the cell expands. And you can see this in the second picture below, which is an autoradiograph. So it's actually showing the distribution of radioactively labeled materials as they're being brought along the hypha. It's a snapshot here. And you can see that those materials are concentrated in the tip of the cell. And these are the structures, these are the chemicals from which the cell wall is assembled. So there's this directional flow of materials toward the tip of the cell. I should mention here, this was based on work by wonderful mycologist called Solomon Bartnicki Garcia, who worked in California before his retirement, very distinguished fungal biologist. So it's showing here the directional movement of materials and the fact that growth is concentrated in particular locations. So we've learned a lot about fungal growth and development from microscopy and also from this biochemical work using these radioactive tracers. I'll show you one more picture of sort of ancient research here, this one from the 1970s, if we go to the next picture. And this shows some really fantastically technically sophisticated work published in the 1970s by Clifford Slayman and his collaborators at Yale University in New Haven. And what Clifford Slayman did at that time was to perform the first intracellular recordings of electrical behavior in hyphae. So what he was doing was putting micro electrodes, so these are glass pipettes that connected to very sensitive amplifiers. He was able to measure the electrical activity within fungal cells, and what he was looking at was the electrical behavior of the cell membranes of fungi. So we get the movement of charged atoms or ions across the membrane of fungal cells, as we have movement of ions across all cell membranes. And this creates an electrical signature. And what Slayman showed in this paper in the 70s was that, in fact, fungal cells produce quite slow action potentials. So we know that on the millisecond timescale, the timescale of thousandths of a second, that we have action potentials that account for the activity in our nervous systems. It's how information flows through the nervous system with the aid of chemical neurotransmitters between nerve cells. And so Slayman was showing here that fungal hyphae, those filaments that form the fungal colony, are also electrically active. And they even have these action potentials, these changes in the potential difference across the cell membrane that follow a very reproducible pattern. We don't know what information is being transferred, if any, here, but it's really interesting from an evolutionary perspective that we have versions of the electrical behavior that we see in higher organisms with nervous systems. We have some kind of primitive version of this within fungal cells. Perhaps this relates to the transmission of information, perhaps not, but it's there. It's another indication of the incredible level of sensitivity, cellular sensitivity, that we find in the fungal mycelium. Let me give you two more examples. If we look at the next slide. What this shows here is actually, so there's a micro pipette, something that we might use to measure the potential difference across the membrane of that fungal cell. So we've got a hypha there, that cylindrical cell that's running through the center of each of these images, and we approach it with the micro pipette. And in this case, when we puncture the cell, what's happened is that it leaks. And we often see this. It's very difficult to actually penetrate a fungal cell with a glass micro pipette or glass micro electrode without damaging the cell. When we damage the cell, we find a pretty sophisticated wound healing mechanism come into play. That fungi in different groups use different systems to actually plug holes when they're damaged in this fashion. They respond immediately, and they have a system that might be compared to the blood clotting mechanism that we find in animals. But they seal off those holes using proteins, sometimes using complicated organelles to actually prevent the loss of fluid. They're pressurized, and so they'll hemorrhage pretty quickly if that damage isn't healed. So we've got sensitivity expressed through wound healing. Last example of fungal sensitivity. Let's look at the next slide. When fungi hit ridges, when they encounter ridges, obstacles in their path, they respond to them. They change their growth form to grow over them, to grow around them. The thing that's labeled there in the middle, it says Spitzenkörper. Spitzenkörper, I think it's German for black body. So it's actually that cluster of organelles, or it's associated with that cluster of organelles. And we see that that structure moves as the cell responds to an obstacle in its path, and indeed it then grows around that a ridge on the surface of a leaf, for example, or an obstacle through which it cannot extend and it has to grow around. And so we see changes in the movement of those membrane-bound vesicles inside the cell that I showed you in that those fluorescence images earlier. The cell is so sensitive, and it uses, I mean, these mechanisms have evolved over the course of tens, hundreds of millions of years. This sensitivity allows the fungus to negotiate its surroundings, to also sense the structure of plant leaves, particularly for fungal pathogens then that need to find openings into those leaves. So they're programmed to sense little changes in the topography, the micro topography of the leaf surface, and to respond to those cues in a way that advantages their search for food, namely in the interior of the leaf. Let's have a look at another picture of, I think the heat's actually gone off. I'm with you today from Ohio, and it's cold, so I'm beginning to shiver at this point, all for the good of scientific communication. These are fungal zoospores. They swim like animal sperm cells. They've got a single tail. A lot of aquatic fungi produce these cells. They're very sensitive to their surroundings. They're capable of sensing the gradients in different chemicals that they swim toward. They show some level of individuality in terms of their behavior. Not every zoospore in a population acts in the same way. So these cells are extremely sensitive to their surroundings, as are all cells. I mean, this is a point I'll make at the end of this discourse. We see we see sensitivity throughout nature. Next picture shows another fungal love of my life, yeast, Saccharomyces cerevisiae. So these cells don't produce hyphae, filamentous hyphae. Well, actually, some of them, many of them do under certain circumstances. But here in the yeast form, they're growing, they produce buds on their surface. And so we can see in the to the right of this image, we've got a mother cell, and she's got the scars on her surface from which daughter cells have broken off. They form by budding, and then they break off and then go off and live yeast lives of their own and produce their own buds. Yeast cells show sort of some behavior that can be compared with the aversion and then habituation to salt that I explained in relation to slime molds. So when we prime yeast cells by exposing them to salt, we find that they become better at adjusting to future stress. So the next time, we give them the pulse of high salt concentration, they respond to this metabolically. There again, this is throughout nature. We see this, whether or not there's any obvious movement. We see evidence of irritability and responsiveness. So I haven't got a vast number of slides to go here. So let's continue though, and then we'll get into some of the more recent work here. This picture just shows the overwhelming complexity of the fungal mycelium. This is just in 2 dimensions on the surface of a culture dish here. A lot of work by some of my colleagues looking at fungal growth and using computer imaging and computer based analysis to try and understand patterns of growth. So these little circles here show where branches are emerging and where where these filaments are actually merging and forming what we call anastomoses, where they're joining, where they're fusing. The pattern of growth that we see in a fungal colony is probably something that's never reproduced. Each colony, at least in terms of the structure that it forms, it follows predictable patterns, but the precise form of the colony is probably never reproduced. This is an individual, so complex once we get beyond 2 or 3 branches even, that this is a unique structure, unique in the history of biology, unique in the history of the universe, something that's only ever formed once. It's based upon an interplay between the fungus and its environment and all these sort of cues to different environmental signals based on the different forms of sensitivity that I've just touched on in the earlier slides. The hyphae are responding to these environmental opportunities and challenges and growing in very specific fashion. Even among clones, you can look at clonal colonies, identical genetic information, and put them side by side, let them grow, and they'll grow in a unique fashion each time. The mycelium is a structure of really amazing finesse, sophistication, complication. Let's look at the next picture. So this is in real time. The red dye here is showing the membranes. You can see the cross walls that are the septa that actually separate what we refer to as individual fungal cells within the fungal colony. The green things are the nuclei. So we've got a multinucleate structure. Each one of those nuclei, well, at least if it's what we call a homokaryon or monokaryon, each of those nuclei are carrying the same genetic information, the same set of chromosomes. And they're moving along the hyphae. Obviously, this little GIF that's repeating here. We have very little understanding of the way that each of those nuclei controls the behavior of the mycelium. They're moving continuously through the cell. Absolutely awe-inspiring, but I like this image because it perhaps shows how little we really understand about the inner life of something as, ostensibly simple as a young mycelium or even a single hypha. So those nuclei have been targeted with jellyfish, green fluorescent protein, jellyfish protein. Okay, let's go to the next slide. Green fluorescent protein. So I'm going to show you in the article. It's open access to you, so you can find it online. Very, very brief read then, where I wrote about fungal consciousness and making this plea to think about fungal ethology, not just fungal behavior or development, but think about behavior from this perspective. Perhaps of even suggesting that fungi are conscious. So let me show you some of the most interesting experiments that have been performed recently. I'll give you a couple of these. So this one doesn't look particularly interesting, but I'll tell you what's going on here. So we've got period 1. You've got a fungus that's growing off a little wood block on the bottom. Period 1, the lower of the squares there is a wood block. We're looking from above, and we can see that a fungal colony, a mycelium, has grown out over that surface. And we've got a bait there. So we've given it a fresh piece of little wood chip, which is uncolonized. And the fungus initially grows out in all directions. When it encounters, it's doing this in a random fashion, as far as we know. When it hits the bait, it begins to concentrate its activities toward that bait because this is a source of food. So it no longer makes sense to keep growing in other directions. At least it makes sense to pour some of its resources into exploiting this source of calories. And the fungus indeed grows from the first wood block to the second. It's not shown particularly well in this picture, but I'll touch upon this again in the next picture. So in the next experiment, what we do is we take the fungus that's growing on the second wood block and we cut it out, use a blade to cut it out from the first culture dish. And then we place it on a fresh culture. So there's no food here. It's a soil mixture. And then we watch it grow out. And what happens statistically is that fungus grows in a direction. In this picture here, it's growing up, right? So it's growing in that direction. Statistically, it's favoring growth in the direction in which it hit pay dirt last time. It remembered that if it grew out in that direction, it was more likely to find food than if it grew out in the other direction. This is a statistical effect. It's not perfect. You can see it here that it's growing also in the other side. But over time, in the researchers that did these careful experiments, they found indeed that the fungus would actually be anticipating a food reward if it grew out in that direction. So this is a simple form of spatial navigation to begin with. How the heck did it actually measure direction? And perhaps we can say this is a very simple expression of intelligence. These are remarkable experiments. They're the first really good ones that have been performed on fungi rather than slime molds that show this kind of behavior, sort of learning, habituation, memory, learning, maybe a form of intelligence. It at least bears discussion. So these experiments, I think, are really important. And the sources for all this work, these aren't experiments that I performed, but the sources are cited in the article that was published in Fungal Biology. Look at a couple more pictures. Look at the next one. I think these come from Mark Fricker, the great scientist. He works at Oxford University in the United Kingdom. They're on resource allocation. Mark really sort of pioneered this work, and you can see here colonies growing out from wood blocks. They're growing out in all directions here. It doesn't show it here, but then if we put a bait in a particular position, and when they find it, they do indeed, as I mentioned in the relation to the previous slide, they'll actually pour their resources, they'll actually concentrate their growth toward that food source. And then they'll score out in all directions again in search of food. They're actually capable of resource allocation too, that once they've found food in one location, they'll actually supply other parts of the colony that perhaps are beginning to starve. They'll move food in another direction, in many directions, to make sure that the colony as a whole is actually well fed, because other parts of the colony might well strike food at any time. And so we need to keep growth continuing in those other directions. The goal here being to what? To access enough resources to put on enough biomass so that one can reproduce. Through reproduction and spore formation, then it sends the genes of this particular fungus down the great stream of time. This is what all of biology does. What is successful persists through time. What is unsuccessful in that respect disappears. Resource allocation. So it's another indication of the complexity of fungi. So a couple more slides then here on fungi, and then I'll sum up with some other comments. Mycorrhizal networks. So fungi, as you know, I'm sure you know, form networks with plants, including forest trees, and we refer to these as mycorrhizal networks. There's been a lot of descriptions of mycorrhizal networks that have suggested that we're looking at some form of natural, the term, the word wood wide web has been used to describe these networks, which isn't a term that I care for. I don't care for it because I think it does a disservice to fungi. The internet is it's complicated, but it's a pipeline for information. It doesn't generate anything new on its own. The internet without information that we input, it's not generating anything new. We can use it very effectively to gather information. It's not responsive to us in a way that, I'm stretching my point here perhaps, but it's not responsive in the way that a mycorrhizal network is. Fungi are creating new networks all the time as they grow in forest ecosystems and connecting with trees. There's far more going on here. Whether we can refer to these as sort of conversations between different parts of the fungal mycelium, it's pushing it, but information is being transmitted from place to place within a mycorrhizal network within forest soil. Information about the nutrient status of trees. So this is a pretty complicated structure. And I think the more we learn about fungal ecology, the more our respect grows for the sheer complexity of these mycelia when they're involved in connecting with other living organisms, plants. So we'll look at the next slide. A couple more to go here. We go too far. I think we go too far when we talk about fungal intelligence. There's a lot of wishful thinking about the way that fungi, and this came long before the essay that I wrote, long before, the idea that fungi are communicating, responding. Yeah, they're certainly doing this, but we're not dealing with the smartest organisms on the planet, but we are dealing with organisms that are capable of transmitting information over long distances. And this is, I would suggest, a simple form of consciousness. If an organism is responding to its environment, transmitting that information from place to place, this seems to me a simple version of consciousness, one that we might describe in those terms. Let's have a look at the next, not generating new information, but nevertheless. If we look at this picture, so I wrote a book a few years ago called The Amoeba in the Room, where I talked about the glories of biodiversity when we look at single-celled organisms, microorganisms. And this just shows the eukaryotes. We're not looking here at the bacteria or the archaea, but we've got these different groups that are shown in the outer part of the wheel there, that these are super groupings of organisms, vast assemblages of life that show genetic relationships. And I'm not going to go into this, the details here. By the way, humans and all other animals fit within that group that we call the Opisthokonta, and that's also where the fungi are classified. So we, as animals, share more in common with the fungi than any of these other groups here. So I've talked about slime molds, well, those sit within the amoebozoans. We've got these organisms called the Haptista and the Crypista, the Stramenopiles. So Stramenopiles include diatoms and giant kelp. We've got the alveolates that include dinoflagellates. We've got rhizarians, archaeplastids that include the plants and the green algae, and excavator. What's in there? Euglenoid algae and so forth. The point of showing this is that we've got examples of tremendous cellular sensitivity in all of these groups, and indeed we see this in the prokaryotes, in the bacteria and the archaea. We see evidence of consciousness, I think, at the level of single cells, something that we might refer to as consciousness perhaps throughout nature. It's a defining feature of life. Arthur Reber has written has written some wonderful articles on the idea of minimal selfhood. What does it really take to be considered a self? Rupert Glasgow is another wonderful writer, has written a number of books. He's based in, I think he's in Germany now. He's written about the individuality of cells and the way that consciousness emerges from perception. And all of the organisms, or there are examples of organisms throughout this great wheel of life here that show tremendous sensitivity to their environment. The ability to discriminate between the helpful and the harmful. Let's look at the last slide. I'm freezing here. 1876, Ernst Haeckel's Pedigree of Man. And so he's showing what are we beginning here? I can't see it on my screen, but the monarans and the amoebozoans at the bottom here, all the way up to the crowning glory of nature then, the great apes and man. At least he's got the other great apes there. And Haeckel actually perhaps didn't see this as emblematic of such an obvious or ostensible hierarchy. Haeckel was very, very interested in structurally simpler organisms. And what's the point of showing this particular slide, which is the last slide? It's just to show, to make the suggestion there that some simple forms of what I would refer to as consciousness exist throughout the tree of life. And I think that the more that we learn about the sensitivity of fungi at the level of individual cells and colonies, the more evidence I think that we're going to find of sensitivity within this kingdom of organisms, the fungi. And I dare to make the suggestion that this might represent some form of consciousness. And that's all I've got for you today as I freeze to death. I'm actually at an office in my university, which is Miami University in Oxford, Ohio, and they have the heat off over the winter, so I'm about to form ice crystal.
Well, I was gonna say we could do some group calisthenics or something.
I need group.
But
bring on climate change. These Ohio winters.
Right. So that was awesome. I want to introduce you to FCP, our house mycologist, Craig Trester, live from New York. Craig, thanks for joining us.
And definitely have to say a big fan of you, Nick. Definitely someone who's wrote prolific about fungi and providing a deep connection. Incidentally enough, I'm holding your very short introduction to microbiology. Cool. I'll hold it up for anyone who's interested.
The fungi
so definitely a fan.
Too. Yeah.
Give a bit in from myself. I'm someone that's very much self-taught and kind of got into fungi because I was interested in environmental remediation, but then went deeper. So a lot of my interest has been like kind of soil emergence and also the interactions in soil between fungi and other organisms, because they're very much in the way we kind of mentioned in consciousness. It's the interaction with the environment, living and non-living. And then we're learning about the interactions with, some of the oldest organisms. So another thing too, and I think since the title was consciousness as well, we kind of tend to view consciousness through kind of our human lens, like an anthropocentric lens, right? When we think about, a lot of our models in neuroscience, for us, we kind of talk about consciousness. The simplest one is the nematode C. elegans, which has 302 neurons, which those 302 neurons, it's still able to maintain homeostasis. And even too, when we grow single neurons in culture, in tissue culture, we can feed them electrical impulses, neurotransmitters, but it's this emergence that happens, right? It's this very puzzling way, like there's kind of, really another interesting kind of clinical psychologist, Zoltan Torrey, wrote the Crucible of Consciousness, but also this brief guide, kind of took this high level approach where, there's three big questions out there. How did the universe begin? How did life begin? And then thirdly, how did consciousness begin? And, we might solve the answer to the first and the second, but the third, a little more complex. And potentially when life emerged, consciousness emerged, right? Because we tend to break down this bias, but, organisms reacting to their environment. And when we look at the structure of our own nervous system, a lot of neuroscientists are, tend to view, like you're a nervous system contained with all these other systems that upkeep and regulate. When you look at the total surface area and volume and biomass of fungal networks throughout the planet, represented as, mutualistic decomposers, opportunists, it's tremendously enormous. And then also the feedbacks and intercommunication between all the other organisms. Kind of one method I talk about with fungi is that, they are the architects of the soil, laying down all that fundamental infrastructure. So just by sheer biomass, when we think about, in roughly a 2-meter person, probably kilometers of nervous cells in our body, we're able to have this emergence in us. What is truly that kind of total emergence that they're doing in the background? Less we anthropomorphize them, but they're doing things. They've been doing this for hundreds of millions of years, recycling nature, laying the form, and we're kind of learning to either kind of engage with them, symbiotically, or continue to engage in a extractive way, which they'll still be here because through every massive extinction event, it seems that fungi have suffered the least loss in biodiversity because of their strategies with relating with ecology overall. But yeah, so definitely that was my little intro blur, but definitely a big fan.
It's interesting, isn't it? The way that, over time that we sort of allow more organisms get to sort of be listed as conscious over time, and I think it's accelerating, right? So if we and perhaps not that I believe in souls, but we got Descartes, right? And the soul is the unitary domain of our species, and then not so much. And then over time, then we're embracing this idea of sensitivity in a greater swath of organisms. But what you said, I think about, the emergence of the first cell. Yeah, that's on some level thinking about consciousness in a way that we can define it properly. It was there with the birth of the first cell.
Well, another thing that's fascinating now is, so as a kind of biology is kind of coming into its own as like a proper science. As a species, we understood chemistry first, then physics, and then we applied those knowledge with principles engineering to get where we are today. But biology is kind of truly coming into its own, largely because of the genomic revolution, which is, had quantum leaps and bound beyond kind of the traditional technological revolutions we've had, whether it's been industrial or electrical to the digital age right now as well. So it's fascinating because we're realizing, this really bootstrapped with the Human Genome Project to kind of build the capacity to interface where we can digitize the code of life into systems that we can look at in this data and kind of do meaningful relationships. But what's amazing is we have this notion, okay, like, one gene, one disease, one link, but it wasn't even that. We're pretty minuscule in capacity. We have roughly, 20,000 odd genes, but, we're anywhere from 1 to 1, our cells to 1 to 1 to anywhere from possibly 1 to 10 or 1 to 100 of our microbiome. We're an ecosystem, and that potentially we co-evolved with, our microbiota, and that we're kind of a mirror of an ecosystem as well. So we think about these feedback loops, right? Largely one of the things that made us human as we are today was largely this kind of very passive influence of the microbes that we made up, that made homes in our bodies. And sought out this facilitation of kind of the almost kind of pre-symbiogenesis, right? This is something we kind of know through history that, as life evolved into higher life forms, it was this coming together based on these stressful events. So it's kind of interesting, like, what is that consciousness, right? Our immune system, especially now, which is kind of coming into importance, which is a very elusive and mysterious part of how we function, but also kind of this interspecies relationships and kind of interdomain relationships that happen within us and outside us, and that it's a continuum of biological matter. One kind of little like phrase that I kind of mentioned with Leighton Morrison, who's another person on kind of this channel talking to soil biology. You ask yourself a question: at what point does the plant end and the soil begin? What plant do we end in the living domain? And we kind of have this bias of, yes, we're able to take materials in nature and fashion them and build things and engineer and construct. But at the end of the day, when it comes to microorganisms, to bacteria, fungi, and archaea, it is more so this process of recycling and extension of their metabolism, which allows for the higher strata of life to exist on. So it's pretty fascinating that we're starting to kind of pull ourselves out of this kind of anthropocentric bias, that we're truly going through this notion of like, that we're more so microbes than we are people, or one-to-one, and then the implications possibly for our future of the planet, but also our health as well.
Yep.
Absolutely.
Short answer. Yeah. I mean, short response. Yes. I agree with everything you're saying. Yeah. It is revelatory, isn't it? To think about sort of somewhere near an equivalent number of microbial cells in our digestive system to cells that contain, human genes. It's
right.
It's remarkable to think of oneself as an ecosystem, and also the way that so much of our behavior is influenced by this onboard microbial planet. Yep. And
yep. Short answer. Yep.
Yep. I don't want to say anymore, but yeah.
Sounds good.
I think, I think semantics are important in terms of people understanding what someone else means when they say a word or a definition, and that it's that's not what I mean, but at least I know what they mean. So between the two of you, I think you've touched on it, but can you define consciousness versus other, like, I think of plants and sensitivity, right? Like, if my plants start aiming their leaves towards the sun and I rotate a pot 180, they'll eventually, go back that way. And I view that as kind of like a sensitivity, not necessarily consciousness. So can you define consciousness, and is sensitivity the word you would kind of contrast it with, or use as something that's related?
I don't think it's inherently something that's definable in a way that where's the cutoff point? Where do we say that this is an example of consciousness on the part of a nematode, but yet the plant's response does not represent consciousness? I'm not willing to say that. I think this is an artificial construct. I think the more that we look at the sensitivity of the rest of life, maybe the more expressions of consciousness that we find. Maybe it was a poor choice of terminology in the first place, because it was so predicated on the human experience, human biology. So I don't have a good answer to your question, but I don't know where that cutoff is.
So maybe we could think about it, a notion that within ourselves, we have our different types of aspects of our nervous system, right? We have our autonomic nervous system, right? That controls our breathing, our heart rate. These are responses, right? When you hop into water, you're going to change your physiology, which is automatic response, which is kind of built into the kind of physiology which helps you survive, right? Things that you don't have to, really think about to do, right? And then there's that's those functions. So when we think about different domains of life, they're responding to their environment, right? And so I guess the question I think we think about with the notion of being sentient, right? Kind of this feedback loop where we're kind of able to experience and think about our own kind of reflection in relation with it as well. It's a consequence of us having a much larger kind of cranial cavity, and also to way more neurons that are dedicated to these different functions. not trying to get into primer of neuroscience and that notion as well. But yeah, it is interesting. Like, if consciousness is a notion of responding to your environment, right? And things that are done kind of automatically, things that are essential, right? For example, let's think about how, every organism on this planet is a ribosome encoding organism. Its function by which it serves to live on a molecular basis by basically taking its genetic information, storing it in a stable way, transcribing it so that it can be read, and then basically actuated so it can serve functions, either structural proteins, functional enzymes in certain ways, right? Is that molecular dynamic? Is that a form of consciousness that it's so baked into it as well? So when you break down to the layers, you kind of wander into the the weeds of kind of philosophy and epistemology and kind of certain notions like that. And the question is, where is that line drawn, right? You could say that a lot of organisms, be it plants or fungi or bacteria, they're far more rooted in kind of the forces of the planet, the sun rising, the diurnal cycles, because they're rooted in it, versus animals. We've kind of incorporated these systems internally. We've even kind of picked up the microbes to kind of outsource this labor as well. And this freed up time and energy metabolism to develop a system that allows to bigger brain cavity, larger functions, and this phenomena of it, right? So trying not to get into philosophy, it's hard to say where that line is. So I guess maybe as humans, we kind of label ourselves sentient, we're self-aware, and we're able to think about it as well. Are the fungi self-aware? We don't know, right? You look at some people,
but
like
I'm not
Andrew
sure.
Adamansky.
I don't see where do you draw that dividing line? So
it's really hard.
It's
yeah. Well, but maybe, and so one example is someone who's more kind of approaching it from an engineering standpoint, Andrew Adamansky, who's putting electrodes into fungi and looking at their responses. A lot of that focuses on how we can engineer with them, but how could we kind of look at those signals that are going on in these networks that are very similar to our neurons, right? How do we have a conversation with a cluster of neurons and a plate, right? How do we have a conversation with 302 neurons that makes up the nematode C. elegans? So it is fuzzy. We don't have a means to do it. I guess in a speculative notion, like, in the future, something like the Matrix, where you could jack in and find a way to kind of have this like lower level functioning, but it's more of a speculative kind of fictional concept, entertaining, right? But I think the one thing I think about is that everything that we've engineered built today started as an idea in someone's head, as fanciful as it was. So it's hard to say, but maybe we can find a way to engineer with biology in the ways we've been crafty as a species so far as well. Who knows?
Yeah, I mean, I think we're looking at a continuum in nature, but of what we might call consciousness. But I don't know. I don't believe in free will. I think that's something that's a philosophical construct. But I doubt that it means much biologically.
If anyone kind of wants to go down that rabbit hole, I definitely recommend to read Chaos by James Gleick,
Yes.
which talks about these dynamic systems, and that maybe the notion of free will is kind of a bootstrapping upon this to kind of ease the uncertainty of it, which I guess in some ways, of people who have enjoyed certain fungal serotonin analogs, you can kind of come at ease with this in certain ways, which is relevant, I think, for a lot of the stress and anxiety and times we're facing in our world today. But yeah, it is hard to draw, but in the notion of understanding that outside of ourselves, the way we built this notion, the way we understood consciousness in a formal discipline, came with this bias of us being the top of the food chain, or being the top, this being our world rather than us being part of the ecology, right? Ego versus eco, and that notion.
That was it's gone off my screen now. That was a really interesting, that comment there. Consciousness, so Jeffrey says, Davison says, consciousness is the ability to choose not to follow common instinct. But at some level, does anything in biology do that? I don't know. At root? I mean, again, with these fungal cells, if I look at a population of fungal zoospores, these are these swimming cells, there's a level of individuality there. I mean, there's a predictability that the way that they'll respond to a pulse of nutrients, but in terms of the behavior of an individual cell there, it's there. Of course, it's predictable.
And that's the mystery of emergence, right? Because out of this chaos, this noise, there seems to be an emergent property. I guess Wolfram kind of looking into like automata and these larger systems, dynamic systems, when you're getting more into like, potentially ways we can kind of look at how like the fine grain of reality functions and all these iterations and tropes, right? There's similarities and these kind of tropes that happen throughout nature and in chemistry and biology and structure, right? So the question is, it's the kind of strange thing. Even going back to like early life, right? People talk about the RNA world, right? Where, how potentially you had a early time and place where, polymer chemistry emerged, right? Where nucleic acids were able to form and potentially there was catalytic function. That DNA potentially might have been a more stable storage mechanism for this early RNA world at the trade-off of loss of your enzymatic function. So it is this weird, right, notion as well. Like, where does you draw the line, from an organism standpoint down to the molecular biology of it? So it's pretty fascinating.
And it's really difficult for us to sort of get over our privileged status for whatever it's worth to think
right.
about it in how do we think about something in non-human terms? Or it's impossible. But yeah, I don't know where the dividing line is. I don't think
I think the closest thing we can do is this is something too. I think as we push forward with information technology and computing, I think the best use of these machine learning and pipelines is systems biology, right? Because that's the way where we could potentially take this huge amount of information and make sense of it and crunch these numbers and get an inkling of what's going on. An intuition at best, right? An intuition at best, right?
Part of it too is a bit of philosophical exercise. It's getting over our disgusting narcissism. Accepting that this isn't the only way to be conscious, right? That my pet cat has her own version of consciousness that, based upon her wiring, is every bit as rich or depressing as mine. It's
exactly. And even other examples of elephants, right? Elephants will migrate, and actually, there's times where it's observed, like in ethology is the observation of animals in their actual natural environment. And these elephants will actually pass an area where a family member had passed away, and they'll seem to mourn, or our projection of what mourning would be. But the question is how we can't physically yet get inside that headspace and experience that qualia. That's the fascinating thing, like the qualia of like, what does that feel like? These different flavors. Where for us is, oh well, surely we're the top of the chain of being. We're below God in this notion. We've been distilled, so we're hopefully, thankfully, walking ourselves out of that kind of egotistical view of the world, and that we're part of it rather than on top of it.
I'm just throwing some comments from the chat.
I thought it was really interesting. Oh, it's gone now. Yeah.
Well, I can throw it back up. You want me to go back to that last one?
Go get with something that really interested me. I'm trying to read them. I thought that's really interesting. Is it one metric for intelligence could be awareness of death and the process of mourning. One could develop that as a definition. It's not an objective way of looking at intelligence, but it's one. Yeah. My pet cat, she's not aware of death. Well, she will be as she, whatever. If she's injured, she's aware of something. I don't know. I'm not making a great deal. I'm not being particularly clear here. It all depends upon definitions.
Well, here's one. So consciousness, awareness and rudimentary memory of immediate surroundings, sensing, aware of self amongst immediate surroundings.
Well, my fungi have all of that, so there we go.
So, Nick, while we have you here, I wanted to ask you a question, something that I've been diving into recently and I think has been picking up. Interesting bacterial fungal interactions that we know that fungi, they have their own microbiome, and then even potentially that the activity of certain fungi, and I think this is pretty important kind of in agriculture, where this is some of James White's research over at Rutgers talking about how potentially fusarium acts as an opportunistic plant pathogen when in absence of its a bacterial symbiont, I believe Pseudomonas. And that I think a lot of the ways that we've interacted with our soils and our biomes, we tend to, strip this microbial diversity. And I think about, I imagine kind of the analogy, if I was in an area where there was plenty of food and environment and lots of other organisms to cooperate with, I wouldn't feel I'd have to look over my shoulder all the time. I wouldn't feel this emergence of I'd have to count things, versus if my environment was decimated, and my opportunity to survive and keep propagating my genes was at risk, that's potentially when I would actually dig into, changing my way of living. So it is interesting too. Like, what are your thoughts upon the kind of new research, or it's kind of gotten more and more out there about some of these bacterial fungal interactions, and even further kind of other interactions within the soil between fungi and different environments?
Yeah, I mean, the more we learn about nature, the discriminating between the whole issue of the individual seems to be dissolving over time. So we see these interactions across nature, and so an individual fungus has its own microbiome, but then those bacteria also are interacting with we're already establishing a hierarchy there, aren't we? That probably doesn't exist in nature. The fungus then has its microbiome. Well, whose microbiome is whose? We're just saying the smaller things are sort of to some level enslaved by the larger organism. But it's
right.
Yeah, this sheer interconnectedness of everything in nature is I suppose for me as a biologist, maybe that's the greatest surprise of my career overall. I haven't thought about it in that way, but I think when I was in college back in the beginning of the 1980s in Britain, we were just, everything was discrete, right? We were, each organism was its universe entire unto itself, and we're absolutely moving away from that picture. This is a larger question, but I mean, the late E.O. Wilson passed away a couple of weeks ago, and I mean, he was interested in sort of documenting every species, naming every species, having this encyclopedia of life, and that seemed to me frankly a rather futile exercise. Why? I mean, for one thing, I mean, why document stuff as we're destroying it? So what? So the aliens can come back and they can look at the mess we've made of nature. But I think also it's more these connections that are more interesting than this almost, to me, a stamp collecting exercise. Anybody that's a taxonomist will send me hate mail, but there is an inherent futility in this as we understand the interconnectedness of organisms, and then even begin. I mean, I find that revelatory myself, thinking of myself and all of my failings as the failings of an ecosystem. It's not just me, whatever I am. Sorry, that probably didn't answer your question at all, did it?
No, that's
I've gone off on a tangent.
It is something that it's a question almost. It's such a new thing. We're realizing it's like a non sequitur because it breaks down our notion of this very linear thought bias, right? We live in a dynamic world, right? And even too, that's that. We shouldn't really think of ourselves as a discrete individual biological entities, but we're just an extension of a continuum, right? I think the closest thing is.
I wonder if there isn't some sort of almost quasi religious wisdom in that too, in the sense of our fear of our own demise, the end of this individual. It's nice, yeah. It's this particular ecosystem isn't going to exist again in the universe, but. I don't know. Is there some satisfaction in that yeah, okay, the ecosystem's going to crash, but I don't know.
Well, I always think about how
I should think about these things before I open my mouth, shouldn't I? I'm not sure.
Well, no, it's good to spitball, but I think about we came up as a species during the Holocene, right? We had this very interesting period in the past 10,000 years, the climate stabilized, right? But in reality, like. We view time as a species kind of at the aspect of like the ecological time frame, right? But it's underpinned by the geological time frame, right? And that you think about like, the biosphere has more so expanded and contracted over time as life reemerges. And that's kind of the beauty of it, right? These dynamic systems that play through evolution and kind of the emergence of it based upon the biotic engaging with the abiotic in different ways. But it's kind of, I think, even further like, in the theory, we've kind of looked at, the holobiont notion, right? That we're a composite, like the larger holo genome. That these.
Can I pause you a second?
Sure.
There's such fascinating comments. It just went off screen. It was something about consciousness not being the same as. Where did it go? I can't. Oh, I can scroll up. There's such interesting comments that are appearing in the the chat here. Awareness. So iMedic says awareness is not analogous to consciousness. I mean, that in itself, I suppose we could talk about that for a couple of hours, but that fascinates me. I mean, this all, of course, comes down to our definitions. I don't know. I think awareness is analogous to consciousness, but I could probably be talked out of that viewpoint. I'm sorry to interrupt. I'm just seeing these comments
No, absolutely.
are appearing on the right, and there's a lot of interesting things here. This is, but anyway, sorry, I digress.
No, yeah, but actually to the point, like we think about ourselves as a composite, right? Like the hollow biont, right? We're an extension of the genes that we have, like the 20,000, and then, there's 100 times as many genes the microbes live on in us, and they're steering our behavior based upon our environment, right? Certain cravings for certain foods based upon the metabolites they're putting out, interacting with our own gut flora and immune cells and gut cells like that. So the question is, it's kind of like we're being steered and guided. So I don't know, it's an interesting intuition, like this notion of consciousness within our own cells, right? Is my immune cell conscious as it works and function with other immune cells to differentiate and kind of present antigens to them? Are my epithelial cells, or my photoreceptor cells in the back of my eye as they respond to different kind of intensities of light? So yeah, it is kind of notion, like we tend to forget, and I try to remind myself a lot that, we're a composite of, I think the number we think of is like 30 to 40 trillion cells, all with the same genome, but expressing different components to function in unison, and, is regulating itself. And then potentially what's regulating us is that this internal consciousness that isn't us, but it utilizes as a home. And then even a notion of how a lot of our diseases we've seen in kind of the modern aspect in the past 50 years, autoimmune, chronic inflammatory, might be influenced subtly by this lack of diversity, how we've impacted it. So it is subtle, right? The notion that you are an ecosystem, that kind of does sit with a lot of people uneasy, as they haven't kind of reflected on the notion of their ego and their own kind of biological individual self-determinism.
I mean, I think it goes beyond this chat today, but I mean, that's a tremendous. It creates some tremendous theological problems for the theologically inclined. I mean, yeah, I mean, yes.
One.
Most faiths, not all faiths. So,
right.
But monotheistic faiths based on the sort of a clear definition of the individual, but not so much.
Absolutely. Well, anyway, Nick, I got to head out. This was wonderful. It was great to meet you.
Oh, good to meet you
too.
Dimensions.
Maybe in one day, 3 dimensions. But yeah, big fan of your work. And if anyone's interested, definitely check out Nick's books. They're a great introduction to microbiology and fungi. So definitely.
Well, thank you. Yeah, good to
happily
good to
recommend.
Good to meet you.
Take care, y'all.
Yeah.
I love that he has them up on the shelf too. He was pulling your books out. But
they're good.
Well,
studies.
why don't we? I think, Nick, I'm cognizant of your time and also the fact that you're probably still freezing. So we could wrap it there. But if you're down to come back sometime, I'd love to talk yeast and other topics that you like to explore. And we have, I think, almost 150 people watching right now. They're all, or I will speak on their behalf and say they're all very appreciative of your time, and as am I.
Oh, I appreciate it. I love the comments. It was sort of frustrating to see so many great comments come up and, not being able to respond to them or fail to respond to them. It's really interesting. I mean, these are a fundamentally interesting issue, isn't it?
So, someone asked about, do you know who Francis Crick is?
I might have heard of Francis Crick. Sure.
Okay. Theory of panspermia. Actually, let me see.
I know of all these people. I don't. Yeah, Francis Crick, of course, one of the co-discoverers of the structure of DNA, and then went on to perhaps even greater things after that. But panspermia. Panspermia doesn't really interest me in the sense that it doesn't really get us anywhere else, right? So life had to start somewhere. I don't think it did, but if it did start elsewhere and then came to Earth on some celestial toboggan, I don't know. Does that change anything?
Someone loves the lapel on your jacket. Can you show us that?
I got this from my sister. It's from the Royal Society for the Protection of Birds in the United Kingdom. So my sister works for the RSPB, and they've got all these wonderful pins, and they've got a couple of fungal ones, and they should really expand their range and do everything. Intestinal worms, and I'd get one of those. I think a tapeworm would be good, wouldn't it? But,
so if you can see some, I'll just put some of the comments.
Yeah, they're great. Hang on, somebody says they'd like to pick your brain with an ice pick with this shit. Laugh out loud. That may be.
Yeah, you can ignore that one.
Mail. Thank you so much.
You see that one, right?
I'm only looking at the nasty ones. No, thank
Right
you.
on
That's nicer than an ice pick, isn't it? So,
I think whatever that was, it got lost in translation.
yeah, it must have done. But I'm sure the sentiment was beautiful.
People are lobbying for a weekly mycology show. Maybe Craig can make that happen. You can be a recurring guest.
Well, anyway, but yes, I'm happy to talk about yeast, our favorite fungus, and of course we are yeast's favorite animal. But there you go. Yeah, happy to do that at some point. And I'm glad we got the technical stuff kind of sorted.
Well, Matt, yeah, just to give everyone background, Nick, his Chrome browser wouldn't allow him to share his screen, so he had to upload his massive presentation. I had to download it, but then when I was presenting, I couldn't see anything on my screen. So that's why when you saw me disappear, I went to another computer and so that I could actually see what was going on the screen.
Right.
But
yeah. Okay.
Yes.
Well, anyway, feel free to email me, anybody. I got a website, themycologist.com, and yeah, I'd love to come back at some point and at least when it's warmer in this office.
Actually, I can post that up. Give me 1 second. Is that accurate?
Yep. themycologist.com.
All right. Well, we will end with that. Thank you, everyone. Thank you, Nick. Again, talking on behalf of everyone, we appreciate your time. And with that, I will kill the live stream. But Nick, stick